Um, can you hear me okay? I can get the meeting started. Please send me in the chat a confirmation that you can hear me okay, then I'll go through the presentation. Hello? Hello, can I, when I talk, can you hear me? Okay, perfect. Then I'll get started. So thank you. It's a great pleasure to, meet all of you and present one of the newest fields in cancer therapy, that is microbiomes in cancer therapy. First of all, I want to thank Professor Dr. Paneerselvam, who was my mentor when I was at Madurai University. And I had the pleasure of visiting some of the students last year when I visited India, and then share a different story with the students. So today we're going to focus on one of the newer topics in cancer therapy, that is also in line with the theme of the A microbes, the International Symposium. And that's why I chose this topic. And this is, the topic that I also worked for a few years, from Takeda Pharmaceuticals. So I'm happy to bring this topic to you guys today. As usual, before that, I usually start any of my presentations thanking two of my, one on the left, Professor G. Shanmugam, my PhD supervisor here at Madurai University. In his lab is where myself and Professor Paneerselvam worked several years ago. He is a great mentor and great human being. And he is in one way or the other a role model for all of us, not only doing science, but also the drive that we have to come back. And whenever I come back, to present to the students in India. And I kind of got the motivation from him because he's one of the professor who gave up his green card and the professorship in US so that he can come back to India and serve students in India, exactly students like me, who got benefited because of the magnanimous characters of the professor G. Shanmugam. Then on the right hand side is Professor David Givol, who was my mentor in Weizmann Institute, Israel. And he, taught me on how to even think about science and do great science. So the one thing that he taught me was it's not the small experiments, the simple experiments you do in the lab that matters. The question those experiments are trying to address, for the community that is the scientific community, or the community that is the community that we live in, and that's what matters, right? So I think the way that he kind of imparted that kind of a knowledge and wisdom is by saying when he did his PhDs, they were trying to address what is the genetic material, is it protein or DNA? So that's the kind of question he was trying to address. So he was a motivation for me to do good quality science in trying to address big picture questions in whatever research you do. So I want to thank both of my mentors before I start my presentation. So today, like I mentioned, we are going to talk about microbiome. Typically, microbiome is nothing but the collection of the microbiota that lives in an environment. And it could be anywhere. The side. You probably in your courses studied about microbiomes in soil, or microbiomes in water, or microbiomes in trees, leaves, whatever, right? So it's a collection of all the bacteria, fungus, archaea, or any microbe that lives in a given ecosystem. And so, that goes without saying. That means there are a lot of microbiome, a lot of microbes that live in our body. And so the collection of those microbes are called microbiomes. And you can see in our body, in different parts of the tissues, also there are a lot of microbes living. The major one being the gut. The stomach has so much, the microbiome composition, and that's critical for not only for our normal health, but also when they get the composition gets disturbed. There is a phrase called dysbiosis, means when there is a disturbance in the balance of the microbiome, especially in the gut, it affects not only one part of our body function, it affects multiple parts of our body function, and that can result in different disease statuses, right? So on the left-hand side, you see the host factors, which are actually affecting the gut composition, microbiome composition. And, it can be, your genetics, the age, male or female, and your lifestyle, how much exercise do you do or you don't, or how clean you maintain yourself, and diet, what you eat, and also the environment where you live in, right? Say, for example, people, when I visited, recently India, and I went to Delhi, where the air was so polluted that you cannot even, breathe normally. So, and as opposed to then when you come to, a village, it's clean air, right? So, and the composition of the microbes in the environment changes as well, based on where you live. And all of these factor in on what is going to be your microbiome. And these microbiome we are doing something to your body, and that's what is in the middle box. It modulates the immune system. It produces a lot of metabolites. It also kind of interacts with, the hormonal and metabolic balance in your body. And it also, helps in a way to maintain, our gut barrier integrity. When all of this gets tilted or affected, then, we might get what we call the IBD, inflammatory bowel disease. asthma, diabetes, or even cancer, right? So today we are going to not talk about all the other diseases. We are going to primarily focus on cancer, how microbiome actually can cause cancer, or how can it affect cancer therapy. And this, in this slide, I'm trying to give some introductory hypothesis on what the microbiota can do to the cancer. Basically, the top top bullet point is just giving you an introduction or the definition for what the microbiota, which is comprising of bacteria, fungi, algae, and protozoa, along with viruses, and they can actually cause cancer. So, and that's what is shown on the picture left side, and the top part of the picture, you see the middle box that shows the carcinogenesis. So some of the microbiome mediated effects can result in cancer formation. But at the same time, that's not the whole story. The bottom portion of the figure on the left shows also different products from the microbiome can actually have an oncolytic activity, meaning cancer killing activity as well, right? And also you need to understand one thing, which is in general, cancer patients are immunocompromised. And we have many different hypotheses. Actually, one of the major in the 50s or 60s hypothesis was the actual cancer genesis, formation of cancer itself is because we get our immune system suppressed. And that concept came because most of the time cancer happens in older patients. And there is very small subset of pediatric cancer. Cancer can be in younger patients. But if you take 80 to 90% of the cancer, it's usually in older age when our immune system is weaker. So the scientists believe, right, when your immune system gets weakened, it cannot do the surveillance enough and remove all the cancers in our body. And that's when the cancer actually starts growing, right? And in addition, let's say, a patient has cancer and then they undergo treatment. So when we give any chemotherapy, all of these treatments also actually affect our immune system and we get heavily immunosuppressed. And when you are immunosuppressed, the cancer can grow happily, right? So, and now if you somehow can activate the immune system, you can fight cancer or you can kill cancer, right? So this is one other way that scientists thought that, we can actually treat cancer. So they found a way to activate the immune system that is suppressed. And that is the recent treatment paradigm that actually changed how we treat cancer patients in the past 10 years, is by treating them with, an antibody called, PD-1 or PD-L1, which actually activates T cells, cytotoxic T cells that can go and kill cancer cells. And also there is another mechanism called CTLA4 instead of PD-1, which is a receptor or an inhibitor receptor for T cells. CTLA4 is another inhibitor receptor that that is present in CD4 cells, and CD4 cells are regulatory T cells that kind of suppress the immune system. Then you inhibit it, then CD8 cells get activated, right? So these two therapies actually change the way cancer patients have been treated. So now any cancer patient that goes to a doctor, this one of the first thing they consider is giving the immune therapy, right? So that shows that if you can somehow activate, that's actually going to be beneficial for cancer patients to fight cancer. So naturally, like we talked about, microorganisms in your body are actually doing something to the immune system. They're keeping your immune system active. So therefore, one way or the other, they themselves can, by activating immune system, can help your body fight against cancer. Or when you give what I call the immune therapy, these microorganisms can boost the immunotherapy activity, so that you can get increased tumor cell kill, right? So that is as a concept being tried, and we are going to see today how those concepts matured, how the scientists attack those problems, and what are the treatment paradigms that are centered around a microbiome for cancer patients. That's what we're going to see today. So this one is just trying to show you, this is like basic um, you know gut cross-sectional view of what the microbes go and do to the gut cells that are in your gut, and what are the immune cells that are getting affected are there, right? So gut is just not doing the digestion, right? So there is a lot more going on in the gut. Remember, gut is one thing where through your food, or by the air that we gulp through mouth, bacteria or microbes can get in, right? So your gut needs to have a protective layer that doesn't let these bacteria easily go into the tissues, right? So that they get shielded and then stay in the gut, and that is done by goblet cells that actually produce the thick mucus layer that is protecting you from not only any bacteria going into your gut epithelial layer, but also it protects you from the acidic environment your gut has, so that the gut will not get slowed down. And also, there are plasma cells that secrete, immunoglobulin A. There are pinnate that secrete, a number of microbial peptides. These are inhibitory peptides, and they go and they try to keep microbes in check. But on the other hand, what happens is the bacteria or the microbes also release some other metabolites, and their what you call metabolic products out, and they themselves go and then interact with the immune system of our body and create a very active immune microenvironment. Basically, the bacterial metabolites, they activate the local dendritic cells, and they produce some other interleukins that can go actually activate the naive T cells into effector T cells. They basically create a very active immune environment, so that the immune system is always awake, and if there is any foreign pathogen that comes in, they're ready to attack, right? So the common salts in the gut actually in many ways not only keep the immune system alert, but also help keep the immune system balanced by activating both activating our cytotoxic T cells, but also the regulatory T cells. In the next slide, this slide is, there are different ways that, we can go intervene using microbiomes in the treatment of cancer. And my goal to put this slide was to give you a few more definitions that, you should know in this field. One is prebiotics, probiotics, and postbiotics. So prebiotics basically meaning food for the bacteria, right? You can think like that, right? It's the fibers we eat, and some of the non-digestible ingredients that we need the bacteria to eat and then use for their growth. So those are the prebiotics. And probiotics are sometimes the bacteria themselves, right? So if you go to the store, there are nowadays probiotic tablets available, right? These are basically bacterial cells that are when you ingest, they start producing new cultures in your body. You can also imagine there are food we take that are probiotics, right? Which is basically any yogurt or curd you take, it's rich in lactobacillus, right? And in the same way, like I don't know if you take kimchi, which is a Korean food, it's also known to be rich in microbes. So those are called probiotics. So you take them, they supplement your gut microbiome. And postbiotics are basically the products from the bacteria. Sometimes it's bacterial metabolites. Sometimes it's the bacteria itself. When the bacteria is dying, it's broken down cell wall, the material from the bacteria themselves. They are called postbiotics. So prebiotics is the food for bacteria. Probiotics is the bacterial component themselves, and postbiotics are the product from the microbiomes, right? So all of these can be used in the therapy, right? And then in the middle is live bacterial products, right? Sometimes, if you identify a particular bacteria is beneficial, there are people that can actually isolate this bacteria and then administer that as a tablet or injection to elicit the function that you wanted to achieve. And also the second is not one bacteria, a synthetic bacteria consortia. So basically, if you identify a bunch of bacteria that may be together or giving better efficacy, then you can give them as, right? So either single bacteria or multiple bacteria. So this is called the live bacterial product component. The last one is, it looks or it sounds obnoxious, but it is very actively being used. But this came not from cancer field. This came in other therapeutic areas. First of all, it was tried in the obesity field, which is called a fecal microbiota transplant, so FMT. So basically what happens is they discovered in the obesity field, there are some patients, you give them however much fat diet, they were not gaining weight, right? And scientists were puzzled for some time, and then they figured out it's because of their gut microbiome. So the bacteria in their stomach is actually helping them not gain weight. So then they tried to come up with a way to, get these bacteria and then give it to people who are obese. So basically, they take the fecal material and isolate the bacteria themselves and purify them and culture them. And now this culture is made into either a tablet, or you can directly inject them in the colon of the recipient. And they found these patients who were obese, now after the introduction of this microbiome from the non-obese patients, became non-obese, right? So and in so people were trying to implement this. If there is a cancer patient that shows a better tumor growth control or better tumor kill, and people are now trying to take the FMT approach and try to give it to patients who are not responding, and they eventually can respond, right? You're going to see some of the data in the next few slides. So these are the ways that the microbiome mediated or microbiome specific ways to actually treat cancer. So this field, like I said, in the last 10 years, this field really exploded. But the original idea and original data that the microbes and immune shaping tumor microenvironment actually played a vital role in cancer, started 20, 30 years ago, right? You can see the earliest data is from, 2002, when they saw the Mycobacterium bovis actually stimulated anti-tumor cellular response in bladder cancer patients. And there are here you see, I've listed a few for which, very nice data available. You can imagine, right? There is a lot of bacterial species that can colonize solid tumors, but only a few that we could identify that play actually a very effective role in actually changing the microenvironment, changing the tumor microenvironment, changing the immune microenvironment in tumors. And some of them are listed here, but these studies focused on a particular type of bacteria way back, right? It's not a microbiome-like study. it's basically a microbe-based study, right? How the microbes are shaping the immune microenvironment for the tumor, right? But today, what we know and study or talk about is how whole consortia of the microbes are affecting the cancer therapy, right? So here is sort of a running list of when this type of research started and where are we today, right? So first of all, in 2007 to 13, right, in those 6 years, the study was only at the mouse stage, right? So there were mouse studies that showed that the gut microbiome can actually, result in anti-tumor immune response, right? It's basically a mouse study, right? Um, 15 is when the pivotal data came, and we are going to talk about, I'm going to show you a couple of examples of these two studies in the next two slides. In 2015, two preclinical mouse studies that showed gut microbiota actually is shaping the immune checkpoint inhibitor responses. So ICI is basically immune therapy. Like I told you, the PD-1 therapy or the CTLA-4 therapy, right? So it's basically you are trying to stop or inhibit the immune inhibitory molecule so that the immune system can be active. So maybe going forward, instead of saying ICI, let's say immune therapy for cancer, okay? So in these two studies, they showed if you change the gut microbiota, actually the mice stopped responding to immune therapy for cancer, right? Now, in 2018, then they started advancing the study from a mouse to human, right? So there, they showed actually, you take the human patient data, the patient who responded and patients didn't respond, and you try to take the FMT from the patients and then try to put them in mouse and then start growing tumors in mouse and then see if the FMT from responding patient is also showing response in mouse, or FMT from a non-responding patient did not, respond in mouse tumors. So this is basically they're moving a human sample into mouse. In 2019 and 20, actually they went ahead and then tried to do that in humans themselves, right? So they were trying to see in humans the gut microbiome is actually playing a role or not in immune therapy. And actually they try to check not only for one type of tumor, they try to check for multiple or different types of tumors. They also try to understand if gut microbiome is more important. Say, for example, if you have a lung cancer, then you should think about lung microbiome. If you have a cervical cancer, then you have to think about a cervical microbiome. So, they started doing studies trying to understand those things. Now, in 2021, there were two clinical trials that tried to do what was done in the 2015 mouse clinical studies. Basically, you take a responding patient and you take their fecal material, isolate the bacteria, and give it to another patient and trying to see if those patients respond or not. So we are going to see the result from all these studies in the next set of slides. So here, I know the next slides, the few are going to be data rich, but let me try to walk you guys through so don't get confused. It's a two complicated slides. So basically what you're looking at on the left-hand side are two graphs. You see the graphs A, B, and C. These are called what we call a tumor growth curves. Basically, on the y-axis is the size of the tumor, on the x-axis is the days. So basically, you take a mouse and you inject the tumor cells, and the tumor started growing over a period of time, right? So that's what you're seeing in A. So in A, it's called SPF, which means specific pathogen free, and B is called germ free. So basically, when you house the mouse need to be housed in a very pathogen-free environment, right? Because when we do the experiments, we don't want the microbiome from the air or surrounding to be a variable, and so you cannot interpret the data, right? So in SPF, which is called specific pathogen free, and germ free is completely germ free. No germ is there in that facility. That means it's a very clean facility, right? So SPF, you can assume only one pathogen is not there, but then other bacteria are still there, right? So you see in graph A, the unfilled circle, ISO CTRL, CTRL meaning control. So that's your control. You see, that's your normal tumor growth. You can see that tumor continue to grow. Whereas if we treat them with CTLA4 antibody, so anti-CTLA4, I told you CTLA4 is immune inhibitor. Now, by giving an antibody to an inhibitor for immune system, now you are activating the immune system, right? So when you activate the immune system, you can see the solid circle, the tumor's growth is slowed, right? You see the control, the tumor continue to grow big. When you give the anti-CTLA4, the tumor is not growing as much, right? But you come to the germ-free setting. Now you see there is no bacteria, right? Nothing whatsoever. In this environment, control and the immune treatment had no effect, right? The tumors continue to grow, right? Which is telling you if you keep it completely germ-free, that means it's not good for cancer therapy, right? In these mice. But you see the graph C here, they are mimicking, it's the same SPF condition, the specific pathogen-free condition. So when you treat control and CTLA4, you should see the same experiment repeated. But the only difference is in graph C, they fed these mice or treated these mice with antibiotics. So instead of keeping it germ-free, they gave antibiotics, basically killing all the bacteria in the stomach. Now they see both tumors from the control and the treated arm continue to grow almost the same way. So the difference is not significant. So basically, this data shows that if you don't have a lot of bacteria, either in the environment or in the body, there is the immune therapy is not working in these patients, right? So what they tried to do was trying to understand what are the bacteria that is present or not present that is actually bringing this effect. But before we go to that, you see on the right hand side, that under the title, ipilimumab induced dysbiosis in patients, right? Ipilimumab is nothing but the name for the anti-CTLA4, right? It's the antibody for CTLA4. So what they found in patients was when you give, this immune therapy, actually the bacterial composition changes. So it's not one way. It's not that the microbiome is affecting immune therapy, but immune therapy is also affecting the microbiome. You can imagine, right? Why, right? This is why I showed you earlier slide. In the gut, there is so much happening, right? Not only the body is trying to protect itself from the bacteria coming into the gut, but the bacteria is doing something to actually keep the immune system active. So if you have your immune system active, you can expect something, will affect also the microbiome composition. So that's what is happening. And they saw. So it's not one way, it's both ways. So here, it's another interesting study where you see on the left-hand side, graph A, B, C, and then E, F, G. So A and E are just like before, right? These are tumor growth curves. The TAC and JAX just mean two different companies from which we get the mice from. So TAC is Taconic, JAX is Jackson Labs. So when they took mice from Taconic and then introduced the tumor cell, and then they followed the tumor growth, and that's what you see the dashed line on graph A. And when you have mice from the Jackson labs, and then you introduce the tumor cells, you see the tumor growth in solid line, right? So you can see mice from Taconic had tumor grow better, and mice from Jackson labs, tumors are not growing as much, right? Then they try to understand why tumors are not growing as much from Jackson lab mice. And that's what you see on graph C. Basically, you're trying to see if there are, interferon gamma spots, basically meaning, do they have active immune system or not, right? You can see in both spot size, meaning, how much they have, and also, how much immune system is active. In both cases, you can see Jack's mice as a higher immune system activity compared to Taconic, right? So Jackson mice that has higher immune activity, the tumor is not growing as much, right? Taconic probably don't have as high as Jackson lab mice in terms of immune microbiome, and so it is growing tumors effectively, right? So now what they did in graph E was basically taking Taconic mice and Jackson mice and put them together in the same cage and do the same experiment, right? So when they did that, now suddenly both mice are producing same type of tumor growth. So what happens here is when the mice are co-housed, now they are exchanging their, spits, their fecal pellet that carries a lot of bacteria. So now somehow when they are living together, their bacterial component or microbiome becomes same, and so they both behave same way. The tumor growth is comparable between them, right? So the one thing you need to remember, Taconic is a lot more cleaner, so the tumors are growing better, and Jackson mice have more microbiome, and so it's highly immune active, right? Now here is where the fecal pellet experiment started coming, right? So I know there are, in 3 different slides, we are explaining. In the first slide, in graph A on the left-hand side, they're trying to show that you can grow the Taconic mice, but if you feed them with Jackson lab fecal pellet, then they started behaving like Jackson mice, right? So that's the bottom line finding. Basically, if somehow you find a way to introduce the microbiome, then they will behave like that animal, right? And not only that, in the bottom graphs, what you're seeing is that also extends to even immune therapy. So if Taconic mice is cleaner and it responds one way, and then you add a Jackson lab fecal pellet, now it will behave like a Jackson mice, and then respond better to immune therapy. So basically this is showing immune system and microbiome are interrelated, and microbiome is critical for immune therapy for cancer. And if you somehow manage to introduce the microbiome from one type of mice to another type of mice, now they will behave very similarly, right, based on the microbiome that we introduced. Then the next step was they're trying to find what is different in these mice, right? So then they went and then looked at their microbiome, and they found the mice that had higher microbiome, the Jackson mice who had a higher microbiome, they found they had a bifidobacterium as the most dominant species. So what they did now is, instead of doing the whole fecal pellet transplant or co-housing the mice, you can take the Jackson mice and then you treat them with immune therapy. You see what the activity was, and then you add them with bifidobacterium, right? Now you added one particular bacteria and then see how the activity was. That's what you see in graph E, right? Untreated is your blue line, and bifidobacterium alone is your purple line, and then the dashed line is immune therapy. But then when you give bifidobacterium and immune therapy, you see the tumor almost didn't grow at all, right? So tumor responded, the tumor actually dying um, when you have the bifidobacterium treated. I mean, bifidobacterium introduced, right? And on the right-hand side, you can see that's because the immune system interferon gamma is one of the interleukin that's critical for immune system. You see in the bifidobacterium and immunotherapy arm, that's the highest. Also, you see the CD8 T cells, like I told you, CD8 is a cytotoxic T cell. And these T cells actually recognize cancer cells and kill them. And you can see the bifidobacterium treated and immunotherapy treated mice actually had the highest T cell, cytotoxic T cell count. So this shows that you can identify the bacteria, and instead of giving the whole fecal pellet, you can give that bacteria. You were able to see the same effect as if you give the whole microbiome, right? So now, these are mouse experiments. Now they try to understand in humans, right? This study now happened in 2018. So this is a melanoma patient study. And these patients are treated with PD-1, which is a different immune therapy, but the mechanism is almost same. It's trying to activate the cytotoxic T cells. So basically, when the patient comes in, you take the initial microbiome from their gut. But here they took both oral and fecal. And then you start your tumor biopsy, and then you start the treatment with PD-1. And then at the end of the treatment, you get again oral and fecal microbiome, and you try to understand what is the difference, right? Now, when they treated these patients, they found some patients responded, some didn't respond, okay? So they call them responders and non-responders, and that's what you see in graph C. R meaning responders, NR meaning non-responders. And what the dots you are looking at is basically showing the diversity of the gut microbiome, right? The more species they have, the higher number, the higher diversity they saw, right? And you see, among the responders, you can see the diversity is very high, whereas non-responders, the diversity is very low. So just on the outset, when they looked at these melanoma patients that had before and after microbiome collected, and they had the treatment, they found patients who had higher gut microbiome diversity responded better, right? Now, then they asked the question, now I have the responding patients and non-responding patients. Now can I see what type of bacteria they have? One is the diversity, right? They had more species present. And second, among them, is there particular bacteria that we can identify that is high in prevalence? And can we use that in treatment, right? So you can see on the figure C here that it's showing whatever is in blue shade, all responders. Whatever is in orange shade are non-responders. And you can see the clostridales were predominant in responders, and bacteroidales were prevalent in non-responders. So this is how they came up with, okay, if one thing is diversity, if you have higher diversity, you will be responding to immune therapy for cancer in these melanoma patients. If you have, in addition to the abundance, if you have, in addition to the diversity, if we had, clostridales, you may most likely respond. And if you have bacteroidales, you may not respond, right? So they were able to even identify. This is called now a biomarker, right? You can identify a bacteria that might predispose you to a sensitivity or resistant to the immune therapy against cancer, right? And here, they identified a species called Faecalibacterium. And among then also they have the bacteroidales. Now this is a slightly different kind of graph. I'm introducing you guys to a different type of graphs used in cancer therapy. So this graph is called the Kaplan-Meier curve. So basically, 100 is all the patients are alive, and then you follow them over a period of time. That's your x-axis. That's the number of days. And y-axis is whenever if there is a patient that's dead, then you go one down, one down. So if um, if the patients live longer, the, the, the line stays above. If the patient dies, then the line dives down fast, right? That means if you want to take the 50, right? See here, if this is your 50 line, you can see in 200 days, right? In 200 days, right? Close to 70 or 80% of the patients are alive in one arm, whereas in the line that is bottom, only 50% of the patients are alive, right? That means in the top one, the patients are living longer, right? So basically, it shows if you look at only the Faecalibacterium cohort, patients who had high Faecalibacterium content, they were living longer. They were responding better to treatment. On the same token, if you look at the bacteroidales, right, the high abundance bacteroidales were not doing well, right? They were dying faster. So you can see there is beneficial bacteria you can identify. There is also not so beneficial bacteria you can identify from this study. Now this is taking the study one more step further, right? We saw already patients responding and they have unique microbiome diversity, and there is certain type of bacteria that's high, they will respond. Certain type of bacteria high, they will not respond, right? So now they wanted to see if that can be now extrapolated or transplanted into another species and same result can be seen. So what you're looking at here in figure G is, you take a germ-free mice, not a germ-free mice, but you take germ-free mice and take the fecal pellet transplant from either responding, or meaning responding, or non-responding human donor, and give them orally, PO meaning oral, give them orally 3 doses to the mice, right, in different groups. Now, after 7 days, the assumption is you give 3 doses. After 7 days, the mice are now colonized by the donor bacteria from the patient who either responded or not responded. Now you inject the melanoma cells, right? Let the tumor grow. And now you treat these mice with the same treatment originally what the patient had, right? If the microbiome is the critical factor for immune therapy response in the patient, if the patient responded, now the mice also have the same microbiome, and these mice should respond to the treatment. And if you take the fecal pellet from non-responder and you fed that mice, and that mice should not respond. So when they looked at, that's exactly what they found, right? So what you're looking at here on actually the graph H is showing non-responders and then the responders. You can see that responding tumors had smaller tumor size because they were responding very well. Non-responders had bigger tumor size because the tumors are not responding, right? And also you can see the same way in the volume too, right? The red lines are growing, they continue to grow, whereas the blue lines, which are responding patients, the mice that had responding patient fecal pellet, the tumors were not growing, right? And you can see the CD8, like I told you, but between the control, non-responding and responding, you can see the responding tumors had higher CD8 positive cells, and that is statistically significant than non-responding. Also, they looked at not only in the tumor, they looked at the CD8 positive cells in the gut, in the stomach. There, the difference was even more dramatic, right? The non-responding tumors had very lower CD8 positive cells, whereas the responding tumors had really high CD8 positive cells. So it shows that gut microbiome from a patient can be transferred to a totally different species, which is mouse, and same response can be reproduced. Therefore, if you take the FMT as a therapy, you should be able to transfer that to patients and then see the same effect, right? So this is, we are making stepwise progress. So here, this study is slightly asking a different question, right? Like I told you, every tissue has its own microbiome, right? Now we are trying to study if you take the lung microbiome, because we saw in melanoma, we saw gut microbiome is related to the efficacy for anti-cancer treatment. If you take the lung cancer, there is a lung microbiome, right? So this study was trying to find out if the lung microbiome is critical or gut microbiome is critical for lung cancer treatment. So what you're looking at here is, again, red is responders, blue is non-responders. And on the top, the Venn diagram you see, this is another introduction of another graphing concept. So the Venn diagram shows actually the microbiota is very different depending on which organ you go with, right? Even from the same patient. If you take the lung microbiome and gut microbiome, they are very different, right? One, there is very minimal overlap between them, right? Second, when they looked at the gut microbiome or the lung microbiome, what they found out was the higher gut microbial diversity is what relevant to response, right? And what you're seeing here is, like I showed you, like I told you, R is responders, blue is non-responders. You can see high diversity, very similar to we've observed in an earlier study. When you have a high diversity, those patients respond better, right? You can see red is response, like 60% of the patients with high diversity responded. Low diversity patients, only 20% responded. So, the 80% didn't respond. So because of the diversity is lower. And the Kaplan-Meier curve, which is D, graph D, also shows the patient with higher diversity. Remember, their graph is above the blue graph, right? So that means they live longer than the low diversity patients. Also, they try to then look for what is the microbial component, right? So you are seeing on the left-hand side, A is gut microbiome, C is respiratory microbiome. Very similar to, you can do the same analysis you did like in previous studies, trying to identify what is the bacterial species. And I told you, in this case, lung microbiome didn't actually contribute much to the efficacy. It was the gut microbiome that, contributed to the efficacy. But they were able to actually identify the specific species that might be, causing, sensitivity to, immunotherapy or not. So here it's a, yet another or a different study. It's a melanoma study in which, in the previous study I showed you in patients, they were treating with PD-1 treatment. Here it is ipilimumab, which is the CTLA-4 treatment. I want you to focus on the left-hand side graph B. You see there are different species of bacteria that was rich in this patient. And what they were trying to find out was which patients, which microbe is related to better activity. So you see the legend for this graph is poor benefit is green and good benefit is blue. So you can see if you have bacteriides, those patients had very poor benefit. Remember back then also we've seen similar with PD-1 therapy. When they looked at, they found out clostridioles were giving sensitivity, whereas bacteroideals were actually associated with a non-responder, right? And you can see here, Faecalibacterium, the second one, you can see more patients are showing benefit and less patients are showing poor benefit. That means Faecalibacterium is associated with response and Bacteroides is associated with non-response. And not only that, um, when they try to find, these patients tend to have inflammation of the stomach or colon, right? It's called colitis, right? So then they try to see if it is associated with a safety or toxicity for the patients. So you can see again, bacteroids are very high in colitis, no colitis patients, whereas firmicutes, which is part of the Clostridioides family that we've seen, they are high in colitis, right? So colitis is an indicator for inflammation, and inflammation is indicator for higher immune activity. And this shows that if we have the Clostridial or firmicutes, your immune system is active, and therefore the patients are actually responding to immune therapy for cancer, right? So now we've heard all these stories, right? There are multiple ways that we can actually try to use this information in trying to come up with treatment for patients for cancer therapy. One way is, like I told you earlier, probiotic supplementation. We can try to influence the immune system. We can try to change the microbiome composition. Prebiotics and dietary fiber supplementation, you can actually try to promote the existing microbiome to just be happy and then and proliferate more. Fecal microbial transplant, we can actually isolate the microbiome component and then use that as a treatment option. Or we can sometimes use antibiotics, right? So if the response is very high, then you can actually mitigate using antibiotics. Or you can also do engineered microbiome, meaning basically using gene editing, you can engineer some of the either the immune system that's actually keeping a check on the microbiome. So you can actually, by gene editing, manipulate that. Or you can actually manipulate the microbiome or bacteria through gene editing and then send it to actually activate your immune system. That's all doable. And people are trying different things, right? So that's on the left-hand side. You can see E. coli Nisi strain, meaning they actually manipulated the E. coli strain. Now you introduce it into patients. Now it's activating the immune system very effectively. So here, this is not only like theory. There are so many clinical trials right now going on in US trying to address, can we actually use the microbiome or impact the microbiome in a way that we can actually increase the response to cancer therapy in patients. And this is an interesting concept that I wanted to introduce you guys to, which is called microbial ecosystem therapeutic, right? It's called MED4. It's an immuno-oncology trial. So what they did in this trial was based on the FMT data I talked about, right? You can take the fecal pellet from patient that responded, put it, give it for another patient. So based on that, what they came up with was, so instead of giving a pellet from a patient, it is if you want to implement that in the actual field, it's not that easy. There is a lot of technical challenges to do that, and you need to scale up, right? Let's say you take a fecal pellet from one patient, and you need to scale up in a way that it will not add to the toxicity, but it will add only to the benefit of the patients. It's not easy to do it. So what they try to do now is they've identified those bacteria, that the composition of the microbiome. So they came up with 30 species of microbial consortium. So they identified 30 species of microbiome, and they also roughly know the abundance, relative abundance each species need to have. And so they actually manufacture this. So this, you don't need to rely on somebody to give you a fecal pellet, right? This, you're creating a consortium microbiome. You're basically creating a tablet comprising of these 30 species of microbes, and do that as part of your cancer treatment, right? When the patients are receiving cancer treatment, you also give this MET4 tablet as well, so that these patients now have the microbiome already altered to benefit the cancer therapy, right? And what you see here on the left-hand side is the result from that study, right? You can see on the left-hand side, so this is yet another graph, and this is called spider plot. You can see it looks like spider legs, right? So you see 100% is the tumor growth, tumor size when the patient actually was treated or started treatment. And if the lines go down, that means the patient is responding. The lines don't go down and the lines continue to grow, that means these patients are not responding, right? So you can see in most cases, you can see more than 50% of the patients actually responding. These tumors are going down, and these patients are living for a year or two now, right? So which is really good, showing in MET4, you see a good benefit. You see on graph B, this is the control. They didn't receive MET4. They didn't receive the bacterial treatment. And those patients don't live, right? You see, they're here only like until 4 months. The longest surviving patient is only like 8 months. But whereas the majority of the patients are done in 4 months itself. Whereas the MET4 receiving patients, they've been living longer, right? And also, what you're seeing at the bottom is this one is basically this is called a waterfall plot. It looks like a waterfall, right? You can see HNSCC, meaning head and neck squamous cell carcinoma. You can see many patients that are below the 0 level, meaning these patients are all responding. And same way with melanoma, there are many patients that are responding, uveal melanoma and genitourinary, right? So you can see in multiple indication, it's not only like melanoma, right? It's head and neck squamous, it's uveal, it's genitourinary. You can see across many different treatments, many different cancer types, excuse me, the MET4 treatment is working. And you see on the graph D, what you are seeing is the ratio between progressive disease versus stable disease, or PR, which is partial response versus complete response. You can see in MET4, the PR to CR, the second middle bar, is high compared to the control where the PR-CR is very low, right? So that shows that with MET4, across all these indications, you see really high response, right? But having said that, right, people try to come up with a biomarker, right? Can we do this consistently across many tumor types and different patients, right? But you can see in this picture, on the A, what I'm showing is, if you look at just surrounding the human, there are different tumor types: ESCC, esophageal, liver cancer, melanoma, lymphoma, lung cancer. RCC is renal cell carcinoma and colorectal cancer. But you see, there are bacteria that are, causing resistance or sensitivity as very different for each cancer type, right? So it's not consistent, and it's different for different cancer types. And so that made it difficult for people to come up with, okay, can we really come up with a treatment or therapy or biomarker that we can use microbiome to treat cancer patients? On the other hand, also, you see now I talked about prebiotics, symbiotics, right? Or the probiotics. There also, there is no like uniform one way or one method that people were able to come up with and try to recommend treatment for patients, right? And that's mainly because the variation among different studies that people have done so far, right? You see, sometimes it's a melanoma study that had ipilimumab treatment. Sometimes it's a melanoma study that had PD-1 treatment. Sometimes it's a lung cancer study. Sometimes it's a liver cancer study, right? So these studies are all different cancer types. These studies are using different drugs. There are different treatments, and every study is using different methodology to analyze the bacteria and also do the sequencing. So because of these variables, the scientific community couldn't come up with like one surefire way of telling patients what to do, what not to do, right? So now what they're trying to do is, can we take, so this graph is showing, you can see it is trying to compute 8 or 9 different studies. And among those studies, if you look at all the responding and non-responding patients, and can you come up with a consolidated view, right? Definitely you can. And you see the blue is responding and sort of orange is non-responding, right? You can see the firmicutes are on the blue side and bacteroides on the orange side. So there are ways that, even in this messy data set that had so many variables, we can come up with some biomarkers that can also tell us in future, can we do a microbiome based cancer therapy that we can, with confidence say it'll work, right? So basically, from, studying all these, we can conclude that gut microbiota plays a role in cancer immunosurveillance, stem cell transplantation, response to immune therapy, and also toxicity immune therapy, the colitis that I talked about, right? And here, another concept that I want to introduce, which is probiotic or synbiotic. Basically, synbiotic is nothing but pro plus, pre. So when you put them together, that gives you a lot of byproducts, and these byproducts do different things in the body, right? And it shows, how not only complex it is. You see the red, these are all basically causing problems, right? They can, the symbiotics together can also, help with cancer growth, can help with tumor metastases, can result in obesity and type 2 diabetes. But also the green ones are the ones that help us, right? So they can do this as well, right? So it's almost like a double-edged sword, and we have to carefully titrate which type of bacteria we need to deal with. And, if the patient can actually, benefit from that type of, intervention or not, right? It need to be carefully analyzed and studied. Here is a kind of a catalog of the postbiotic products, right? We did talk about postbiotic products are the byproducts of the microbiome, or the the bacteria, the viral, their component itself, right? You can see, if you look at the postbiotic products, I told you from the symbiotics, you can see there are so many, it can go good way or bad way, right? Whereas when it comes to postbiotics, except one, right? And even in this case, this is not bad. Everywhere it is actually beneficial, right? We are trying to induce apoptosis against cancer cells, or we are inducing cytotoxicity, which is cell kill of cancer cell. So most cases, postbiotic products, they're all beneficial, right? Whereas a symbiotic product or a probiotic products can go either way, right? And here, in summary, that microbiota, like we talked about earlier, it can actually modulate not only adaptive immune system, because we talked so much about primarily the CD8 positive T cells and CD4 positive T cells, but it can actually impact innate and also the tumor cross antigens as well, right? Innate, I think, we talked about the first slides. It does impact the dendritic cells. It increases the NK cells activation. It increases the monocytes. So these are all the innate immune system components, right? In adaptive immune system, we already saw that Bifidobacterium or Faecalibacterium, they actually activate the cytotoxic T cells that can go or kill cancer cells, whereas the bacterioids, they actually activate the CD4 positive T cells, which are called regulatory T cells. They go suppress the tumor cell killing T cells, right? So they basically inhibit tumor cell kill. And also, tumor cross antigen presentations that can happen because of microbiota, right? So this is my summary, where gut microbiome, actually it's a very complex community. with various features. And these features, they just not only mean the diversity in the community structure, the abundance of each, with the taxonomic composition, which means the abundance, and also the metabolic characters of the microbes that are present within the microbe, right? So the microbiome is very complex, right? Just the gut itself. But then, we also saw different microbiomes. They're also very different from each other, even from the same patient, right? And pre-immunotherapy, anti-cancer treatments we talked about, they actually modify or modulate microbiome, right? I told you the gut microbiome modulates immune system, but actually our treatment, the cancer treatments, or any other antibiotic treatment that we take in our body, it alters the gut microbiome actively. Immunotherapy or anti-cancer treatment, it causes dysbiosis, and baseline gut microbiome can not only predict the response, but also it can predict the safety events, right? So we talked about how colitis can be predicted, and it's because of the immunotherapy with the gut microbiome, right? And we also saw the diversity and abundance of the gut microbiome is a reliable biomarker, right? Irrespective of the tumor type that we went, and in the studies we saw lung cancer versus melanoma. Also, we saw a study that kind of went cross-sectional across the head and neck cancer, uveal melanoma, and actual metastatic melanoma, and also genitourinary tumor. So across the tumor types, gut microbiome is a potential a biomarker, and also shows that longitudinal sampling, meaning in human studies where you take the sample, you do the treatment, and then you stack the sample again, and when you compare the microbiome, there are only minor changes because of the treatment, but overall, from the same patient who's responding and not responding, it's very stable, right? And the responding patients have responding microbiome, and non-responding patients have relatively stable non-responding microbiome, right? And we also saw that you can alter the gut microbiota to manipulate the efficacy for immunotherapy for cancer, either through prebiotic supplementation or even the fecal, pellet, transplant, or using antibiotics. You can modify the immune therapy to cancer, right? So this field is still evolving, but I want to finish the presentation with this note. If you don't know, right, Jennifer Doudna, actually she's a great scientist, and she's the one who discovered CRISPR. And she recently had her book published 2 years ago, and she is actually a major player during COVID time in trying to curtail the pandemic. But she is taking microbiome as her approach to actually not just for cancer therapy. She's trying to take microbiome manipulation to solve immune disorders, right? Autoimmune diseases, as well as mental illness. And she thinks that we can take and change microbiome and bring even climatic change, right? So, and she is thinking big, and there are bigger and better things to come. Please keep on the lookout for this space. So with that, I want to thank you all for patiently listening to the presentation. I hope you not only enjoy the presentation, but you had some things that you didn't know before. So with that, I want to conclude and thank you. Please applaud yourself if you guys are in the room for listening to me patiently. And if you have any questions, I don't know if there is a way to interact. You can either ask through chat, or you can come to the mic and present and ask questions. Thank you.
With the deep up With deep appreciation, we would extend our sincere gratitude to our eminent speaker. Now I invite Ms. Priyadarshini, First MSc Department of Microbiology, to propose the vote of thanks.
Good morning to distinguished delegates, eminent speakers, and esteemed participants from across the globe. As we conclude this session, it is my honor and privilege to propose the vote of thanks. We are deeply grateful to Dr. K. Kannan for his outstanding and enlightening keynote lecture, Microbes and Cancer Therapy. His presentation offers a comprehensive global perspective on a role of microbial science in advancing cancer therapies and highlighting cutting-edge respects with significant translation impact. The depth of insight and clarity with which he addressed this complex topic greatly enriched the scientific deliberation conference. We sincerely appreciate Dr. Kannan for sharing his invaluable expert time, international experience with us, which has undoubtedly inspires meaningful discussion and future collaboration. I would also like to extend my sincere thanks to the organizing committee, scientific advisory board, and all collaborators who dedicates efforts to ensure the success of this session. My gratitude is also extended to all the delegates for their active participation and thoughtful engagement. On behalf of the organizing committee, I thank each one of you for being the part of this precious international conference. Thank you.
Now it's time for tea break. Refreshment will be served outside the hall. After the break, oral presentation will be conducted in the NR hall. Everyone are requested to be present at the hall at 12 p.m.
time, expensive equipment, and complex reagents, and the necessity of trained personnel. So World Health Organization recently announced 12 bacteria. That 12 bacteria is the multi-drug resistant bacteria. Among 12, the Pseudomonas aeruginosa is number 2. And also our Prime Minister has addressed antibiotic resistance is a serious threat. It has been affect our nation health and as well as our nation economic burden as well. Next. So, define of electrochemical biosensor. So, it is a combining of biological recognition element and electrochemical transducer. So, which is comes from electrical signal to the electrode. So, what are the components main important of electrodes, transducer surface, and bioreceptor analyte. So, analyte nothing but pathogen like bacteria, virus, and so on. So, what are the advantages of these techniques? Portability and adaptability. So, I have chosen the optomer. So, optomer is the synthetic recognition elements, which is their tunable binding affinity and long-term stability. So, that's why we choose an optomer for this study. And we chosen the two objectives. One is electrophoretic deposition, which is this platform to immobilize on methylene blue optomer as a redox probe for Pseudomonas aeruginosa detection. And second one is amine functionalized, both side 5' and 3'. We designed the amino functionalized group. So this optomer, it's like a self-oriented binding affinity. So that's why specifically these techniques detect only Pseudomonas aeruginosa. So these schematic illustration of fabrication and detection principle of carboxylic multi-wall carbon nanotube we chosen, and optomer based electrochemical optosensor for Pseudomonas aeruginosa. So next. So experimental details, we have chosen electrophoretic deposition and biosensor fabrication and electrochemical detection of Pseudomonas aeruginosa. And this sequence is the oligonucleotide, it is called optomer. And what is the dissociation concentration of after fabrication? 17.27 nanometer. So this is the results. We confirmed FESM for multi-wall carbon nanotube is present or not, and we performed a Raman spectra also. And after methylene blue optomer conjugation, we tested FTR and XPS analysis also. So those characterization has confirmed optomer is binding. Next. So this is the same image before and after binding Pseudomonas aeruginosa, and AFM also we confirmed in the electrode surface, and we tested a step by fabrication of electrochemical sensor like bare graphite and multi-wall carbon nanotube, and also we test that is cyclic voltammetric analysis, and figure P is the impedance spectra. We confirmed how the electron transfer and how the material has effect to stability, and also we tested square wave voltammetry for the stepwise fabrication, and also we tested Pseudomonas aeruginosa from 10 to the power 1 to 10 to the power 7, and we finally tested different frequency, because frequency is the main role of sensing aspect. 25 to 80 frequency hertz, and also we test incubation time and temperature and different number of electrodes and different number of pH. So pH 7 is the optimum pH for this sensor. Next. And also in these images we tested undiluted urine without treatment. We just drop the modified electro platform, so which is how it is working on the device. And also we tested different kind of measurement of sensor, sensor 1, sensor 2, and sensor 3. So finally, we did imaging studies of Pseudomonas aeruginosa, fabricated electrochemical device, and real matrix. So our concept of disposable baby diapers and sanitary napkin. In future, we develop a in vitro urine monitoring of smart diaper and sanitary napkin. Next. So these are the cross reactivity of the different bacteria. So we tested a number of strain, particularly Pseudomonas aeruginosa and Pseudomonas aeruginosa PA01 is the target of this platform. Next. So our summary and conclusion. So developed a simple and highly sensitive electrochemical apta sensor based on the methylene blue labeled aptamer for selective detection of Pseudomonas aeruginosa, and employed square wave voltammeter the binding molecules, and achieved while linear range of 1 into 10 to the power 1 to 1 into 10 to the power 7 CFU. And finally, ultra low detection limit of 1 CFU, and demonstrated excellent sensitivity, specificity, repeatability, and operational stability. And we validated practical applicability of direct detection of Pseudomonas aeruginosa in undiluted human urine. So established proof of concept real world use under stimulated urinary tract conditions using disposable absorbent materials like baby diapers and sanitary birds. These are the future studies. So these that graphical image is the final output this platform. Next. Thank you.
Thank you, Dr. Bhubalan, for your nice presentation.
Thank you, sir.
Now the presentation is open for discussion. Any queries or clarification from the audience? Anybody want to ask something? You have to interact. Then only you can get something, or the speaker also have. Is it a diagnostic test, or so what kind of your invention? Is it a diagnostic kit?
Sir, future we are going to implement the portable and affordability for the diagnostic platform.
Okay.
So first we designed the screen print electrode. In the screen print electrode, we design and developed the material based. So we have chosen the SELEX is the computational based studies. So we orientation we checked, and based on the orientation, the sequence oligonucleotide. So that oligonucleotide it's self-oriented in the bacterial nucleotide. So that's why specifically Pseudomonas aeruginosa capture this platform.
Oh,
In
nice.
future, we developed for the sanitary napkin and urine child diapers. So we already running on that, and hopefully it will come up.
Any preventive methods for that avoid of this Pseudomonas aeruginosa infection?
Sir, it is immunocompromised pathogen, as you know very well. So we should aware healthy and hygienic. That is the main impact. The Pseudomonas aeruginosa, it will affect water also.
Yeah.
And wastewater also, it's outbreak. So we are very cautious to
So
keep on.
opportunity pathogen.
Yeah, definitely.
Okay.
Thank you, sir.
Thank you.
And also, I would thank organizing committee, but this is not enough for this presentation. I know, and also I am the anomaly of this
Oh, nice.
college. So I did BSc Zoology, but 1 year I had here. Then I moved from Saurashtra College, where there I did PG and UG, and then I moved IIT Madras, so on US, and now I got the award from National Postdoctoral Fellow. Still, I am working. Thank you for opportunity.
Thank you.
Thank you. The next lot number is 2. Lot number 2, Aruna Devi. Stick on to your time. The time for your presentation is 6 minutes and 2 minutes for the discussion. Your time starts now.
To all, from Tyagarajan College, Madurai. Today I talk about my work, mushroom cultivation used Rhizonia regia. Then introductions. Royal palm leaf sheath is a visible and abundant agricultural waste product that can be utilized as a lignocellulosic substrate for mushroom cultivation, particularly for oyster mushroom. The next slide. Then objectives. Some objectives are included: economic empowerment, waste management and sustainability, food security, nutrition. Economic empowerment, creative self-employment and profitable small business. Waste management like paddy straw, then other wastes. Food security and nutrition provide a protein-rich or vitamin-packed food source helping to combat malnutrition. Next slide. Next slide. Then methodology. Methodology of royal palm sheath mushroom cultivations. Substrate preparations, moisture adjustment. Substrate preparations, I collect royal palm sheath, fresh royal palm sheath. Cut the small pieces to reduce particular size. Moisture adjustment, add water to achieve an optimal moisture level. The substrate should be moisture but not waterlogged. The next slide. Then next method, sterilizations. After cut these pieces, I have sterilized. Oyster mushroom pasteurization is often sufficient and involves heating the substrate. 60 to 80 degrees Celsius, I have sterilized. Here 1 or 2 hours, because most harmful microbes are killed in this process. Then inoculations. Inoculations are sterile environment like laminar flow and near flame. The mushroom spawn the seeded mycelium grow on grains mixed into the prepared substrate. The next slide. Then incubations. The inoculated bags are placed in a clean and semi-dark room. Allow the mycelium to colonize the entire substrate. Then conditions 22 to 25 degrees Celsius for oyster mushrooms. Durations, full colonization usually takes 15 or 20 days, indicated by the substrate turning uniformly white with mycelial growth. The next. last stages of fruiting and harvesting. Then fruiting initiations, the bags are exposed to light cooler temperature. Humidity is increased 85 to 95%. Then after bags are holes and cuts may be made in the bags to allow mushroom development. And last it says harvest. The mushrooms grow and are harvested in several flushes or cycles. The next slide. Then first process, cut the palm sheath soaked in 24 hours. The next day, dry the palm sheath and shadow places. Then after palm sheath are dried, bags, palm sheath are filled the spawn bags. The next slide. That I concluded using royal palm sheath and palm waste in general is an effective, economical, visible and environmentally friendly way to cultivate mushrooms, particularly oyster mushrooms. Results are going. And thank you.
Yes, sir. Okay. Thank you. Mushroom cultivation.
Yeah, I will try.
Actually, the mushroom cultivation, one thing is recycling waste. That is, you have to select any waste. But royal palm, where did you collect that royal palm?
It's available in my. So I have tried.
Fresh specimen you have collected and put for substrate.
Fresh collected, I have try panni.
Okay. Mushroom and royal palm.
Results going, sir. So
It's going on. Just you are tried.
Yes, sir.
Okay.
Yes, sir. So thank you, sir.
The next lot number is 3. Nisha, sorry.
Good afternoon to all. I am Nisha from Tyagaraja College, Department of Botany. So today I am going to present on my work, comparative study on oyster mushroom cultivation using selective padistra substrate. Next. Coming to the introductions. So oyster mushroom is one of the most commonly cultivated edible mushroom. Because of it is a high nutritional value and simple cultivations method. In India, mostly the padistra is cultivated or generated every year, but most of it is unutilized. So it is eco-friendly and then easily degradable. This is my objectives. So to compare the growth and yield of oyster mushroom to different padistra substrate, to analyze the morphological parameters such as type, length and weight and yield, to analyze the nutritional content, ash content and lipids and fat. Next. So this is my methodology. 2 substrates were selected. Then the name is a traditional rice variety, which name is Ratasali. Then another one is commercial rice variety. Its name is RNR. It is easily available for me because my father has cultivated our own land. So that's why I'm selecting these 2 types. So paddy straw was cut into the small pieces and then soaking in water in 16 hours. Finally sterilized. Then for packing and then add for spawn uniformly. Finally incubating the mushroom cultivations room. Then next. So this is my result. So comparative study. First one is commercial paddy straw. This is the paddy preparation of paddy straw. Then packing. Finally got it. Then is traditional ratasali, paddy preparations and then packing. Next. This is the mushroom growth rate. First one is the tool of it is a white color. It's a tool spawn is traditional paddy straw. Easily analyzed. I'm put so previous please. So next is fully colonizations. And then 15 days colonization of commercial paddy straw, then 17 days of colonization in traditional paddy straw. So comparatively analyze. So 2 days is a very 2 days is closely growing on. Then says finally the fruiting develop. 25 days of mushroom fully developed in commercial variety because easily penetrated. So that's why 25 days let's grow on. Then traditional paddy straw is 27 days because the ratasali paddy is a very strong and then fiber. So that's why not penetrated, easily penetrated. So 2 days extra. The next. This is the commercial paddy straw. Then is traditional paddy straw mushroom cultivations. The strip length is long and then fruiting body's weight is long. Weight is high. Then compared to the commercial paddy straw is lower. Next. So nutritional analyze. I have analyzed on 3 kind of content. So ash content and fat content and lipids content. Next. So 5 gram of a sample was taken, then hotter oven, heated in hotter oven. Then finally sample is cooled and then wait for sample. The ash content is calculated. Next. This is the graphical representations. So violet color is represent for the traditional paddy straw. Then red color is commercial paddy straw. So traditional paddy straw high ash content compared to the commercial paddy straw. So next. So this is the fat content. So 10 gram of mushroom samples was taken. The in petroleum ether extractions was in previous please. The petroleum ether extractions incubated, then finally the extracts was filtered, then cool it and then wait it. Next. This is the fat content. So traditional paddy straw is a percentage of 70% and then commercial paddy straw is 43%. Next. This is the lipid. 5 gram of sample taken and then chloroform and methanol was added in ratio for 2:1 ratios. Then mix swelling, kept it in 3 days and finally take it and filter it. Then centrifuge for 3000 RPM. The upper methanol layer was removed, then chloroform layer was evaporated in hotter oven. Then finally crude lipids were taken. Next. So this is a graphical represent for the lipid content. So traditional paddy straw is high lipid content for compared to the commercial paddy straw. So mostly the mushroom cultivation is less fat content. So it's a similarly 47.31, the 44.89. So it is a 3 point is high for traditional paddy straw. Next. So this is my conclude. So traditional paddy straw longest type and higher fruiting body and then weight and high yield. Commercial paddy straw substrate produced moderate yield. Then nutritional analyze. The traditional paddy straw showed higher ash and fat and lipid content. So indicating the better nutritional quality compared to the commercial paddy straw. So that's it. Thank you.
Thank you for your presentation. Why do you select different varieties of paddy straw? Because you have how much variation you have seen. The analysis, phytochemical analysis for paddy mushroom.
Sir, paddy straw are two kind of varieties, sir. The traditional one is, another one is commercial variety.
Okay.
The name is Arana and Ratasali, sir.
So what about the yield? Because your objective, you have to calculate the yield of mushroom. Anything you have, your
Sir,
result
not
is
mentioned that, but traditional paddy straws one bag, first time yield for 150 grams. So compared to the commercial variety is 75 grams, near 75.
Okay. So you are more yield from
Traditional paddy straws.
traditional paddy straw. Okay. So actually mushroom which depend on the substrate. What chemical observe from the substrate? The mushroom growth, your yield, what kind of nutrition in the substrate?
Sir, iron.
Iron content. So you have analyzed ash content, fat, and what
Sir,
for that?
Commercial and traditional.
What is the contribution of fat in mushroom growth? You have analyzed, no? Paddy straw, you have analyzed fat. You shown that result. Ash content.
Yes, sir.
What are their contribution to mushroom growth? That lipid content, fat content, differentiate lipid and fat.
Sir, different methods are analyzing, sir.
So what is lipid, what is fat?
Sir, traditional compare பண்ணும்போது traditional better இருந்துச்சு sir. So thank you, sir.
The next lot number is 4. Your time starts now.
Good afternoon, everyone. So my topic is about fabrication of eco-friendly bioabsorbent materials for the napkins and diapers, which is made upon cytosine and cellulose. So before entering into my topic, I would like to discuss about these things. These are the things, row of infection, which is caused by improper use of sanitary napkins or prolonged use of the diapers and so on. Such as rashes, UTI infections, and vaginosis, even to the hormonal imbalance, which leads to the chronic conditions like cervical cancers. So, next slide. For decades, the global is evolved with the usage of the commercially available napkins, which is fully loaded with the polystyrene things like polyacrylic gels, which hold the moisture from the menstrual blood and so on, which is prolongedly used by ourselves, which cause a series of infections such as the rashes and even severe more infection in population. In group of 10 members, now more than 4 or 5 people are affected by the hormonal imbalance such as polycystic ovarian syndromes and disease, which is not shown by any symptoms. It is only noticed and diagnosed after the chronic state was attained. So my objective of the project, next slide, is to design and fabricate the most eco-friendly and biodegradable bioabsorbent containing pads and diapers, which will serve for the mankind. Then the components which I chose was the chitosan and cellulose. Chitosan that I was extracted from the organism of swimming crabs. We all noticed that crabs in the local markets which had the shells with 3 dots, that was known as Portunus sanguinolenthus. The chitosan yield is high in that organism, so I chose that organism for the chitosan extraction. And the cellulose was extracted by vetiver grass. I chose vetiver grass for its antiseptic and cooling effect. We know that in every cycle, the women was affected by the period rashes and some itching sensation. It is due to the high heat and prolonged use of the used napkins. With the help of the vetiver grass, we will overcome that issues by its cooling property. Then methods. So the series of methods was the extraction of base materials such as chitosan and cellulose and optimizing the yield, on which point we can have the much of yield with the low quantity of raw materials, and characterizing the process. Then the main process is fabrication. I have planned to fabricate the absorbent material as the 3-layer of bed, like unwoven wool or cotton material, and inside the bioabsorbent which I was produced, and I have planned to do the degradation test, because degradation is most important in bioremediation processes. It is the process. The process goes on like this: the extraction of the base components and combining the material to form the web-like mesh to absorb the liquid particles due to the period cycle, and the final absorbent is produced further studied. We are in the initial step of extraction and characterization process. Then the product benefits are active antimicrobial protection, zero rash breathability, toxin-free menstrual care, waste to well sustainability, and ability rapid circular disposal. So these are the reference. Yes, that's it.
Thank you, Ms. Visalakshi, for a nice presentation. Actually, how do you isolate that chitosan from the organism?
Yes, sir. We initially collected the exoskeleton of the crab, which I was choose the organism proteinous, and dried it over 2 to 3 days in hot air oven at Celsius in order to avoid further more destruction in that. And before that, I have removed all the flesh, because most of the people in a group have the fish and aquarium related allergy, so I don't need that. In the process, I do the deproteinization, demineralization, and deacetylation step. So the protein was removed. Only the absorbing material which I want is isolated, sir. That's why.
Is it bind with other cellulose?
Yes, it combined with the cellulose.
What kind of binding did you? I think what kind of binding is there with chitosan and cellulose?
Sir?
So what kind of binding will be there in the cellulose content and chitosan?
Sir, we have planned to use the cellulose that was obtained from vetiver and the with the use of glycerol like thing. So
No, any specials for
no,
this?
sir. No, we didn't.
No.
We didn't test
So
on it.
how it will be stability will be there on after binding with the fabrication? These substances, chitosan and cellulose, how it will be bind on that fabricated matter?
Sir, initially I have planned to make it like the film-like thing. So it will like the rigid compound like. No, sir. The protein which was present in the was a tropomazone. It was eliminated due to the deproteination process. So the soluble matter alone extracted from that.
Why you specifically chosen this? There are so many shell organisms from which you can extract.
Ma'am, comparing with the blue and red crab, I initially choose that. But the yield of chitosan is comparably high in the proteinous variety.
So how much do you collect and get the?
I initially collected the 2 kg of exoskeleton.
2 kg you need.
2 kg I collected, but I make it into the powder form. I get only 500 grams. So I do the steps like 15 initially. The crude chitosan powder gives about 5 to 6 grams of purified chitosan. Thank you.
The next lot number is lot number 10. Stick on to your time. 6 minutes is for your presentation.
Good afternoon, honorable judges and their delegates. I am V. Prabhakaran from Tyagarajar College, Madurai. Today I am pleased to present my work titled Sustainable Mushroom Cultivation Using Groundnut Waste as an Alternative Growth Substrate. This studies focuses on converting agriculture waste into valuable food resources. Next slide, please. First, we see about the introduction. Mushroom cultivation plays an important role in global food security by converting lignocellulosic waste into high quality protein. Currently, the mushroom industry faces the mushroom industry faces mounting pressures to adopt sustainable practices while maintaining economic viability. Agro-industrial waste streams offer an unexplored potential as a cultivation substrate. The agro-industrial waste has been transformed into a productive resources. This approach addresses both waste management challenges and growing demand for an alternative substrate material for mushroom cultivation. Next slide. Challenges in the current substrate. Currently, we we use rice straw, wheat straw, and sawdust for the cultivation of mushroom. I cultivated the mushroom in groundnut waste cells. What are the challenges in the current substrate? Conventional substrate limitation. The current substrate has a face available constraint and rising cost, regional shortages, and then the environmental impact of agro waste. And the used agro waste has been burned and disposed in the landfills. This practice release greenhouse gases and create public health hazards. A need for diversification. Most of the mushroom cultivation has been cultivated in rice straw, wheat straw. We also need a different type of substrates for diversification. Next slide. Groundnut waste, an untapped resource. Why I choose groundnut waste? Because there's abundant availability and having futures in the other traditional substrates like rich in cellulose, semicellulose, and lignin, essential compound for mycelial growth. Next slide. My studies aim and objectives. My studies mainly aim to evaluate the growth in a groundnut waste as a primary substrate for cultivating edible mushrooms and comparing growth parameters, yield metrics. The objectives of the studies include mycelial colonization rate, yield performance, and biological efficiency, and optimal processing methods, and evaluate economic feasibilities to marketing to the small scale farmers. Next slide. Materials and methods. I use this Pleurotus ostitis for the cultivation. Groundnut shells has been collected and dried and ground uniformly. Substrate preparation, and the moisture adjustment 65 to 70%, supplementations with the nutrients, pH optimization. This groundnut cells has been autoclaved at 121 degrees Celsius for 90 minutes. Incubation conditions, it has been incubated in room temperature with a humidity of 80 to 85% with controlled light exposure. Next slide. These are the evaluation metrics I has been studied in my work. Mycelial colonization rate, span run, duration, yield assessment, biological efficiency. I forgot about tell one thing. My study is an ongoing process. Next. Preliminary and observation and anticipated results. Currently, my work conditions in the week 3. First, I inoculate the species in the substrate. Mycelial growth has been visible for from 5th day, and then the week 3 progressive mycelial growth has been seen, and the anticipated result will be we expected to get a better yield perform. Next slide. Significance of the study: environmental benefits, waste valorization, economic feasibility. Next. Thank you.
Thank you, Mr. Prabharan, for your presentation. So now you are doing on your project on that mushroom cultivation on
groundnut waste.
Groundnut waste, very good.
ஒரு groundnut வந்து யூஸ்ஃபுல்லாக அது இது வந்து வேஸ்ட்டாக தான் இது பண்ணுவாங்க. லைக் வந்து ரைஸ் ஸ்ட்ரா வந்து அது வந்து வேல்யூ இருக்குது, பட் கிரவுண்ட் வேஸ்ட்டுக்கு வந்து வேல்யூ.
பார்த்துருக்கீங்களா கிரவுண்ட் வேஸ்ட்டாக எரிக்கிறதையும் பர்ன் பண்ணுறதையும் பார்த்துருக்கீங்களா? ஷெல்ஸ் தான் பார்த்துருக்கீங்களா? No.
Okay, sir.
Groundnut waste, actually this is not a waste. We can alternately use anything. Okay. Very high energetic substance, anything. It's okay. Your preliminary observation.
Thank you, sir.
Thank you. The next participant is...
So, wait. So, the identification of the poop in GCMS analysis, the three marine microalgae species for the linoleic acid, alpha-linoleic acid, and eicosapentaenoic acid, the tocosahexaenoic acids. Next. I have selected the classification, and I have selected the isocracy species. It is a brown golden marine algae. It is a flagellate microalgae with a ovoid shape, measuring 4 to 6 meters per diameter. High content of the polyunsaturated fatty acids such as tocosahexaenic acid and stearinic acid and alpha-linoleic acid. Next. Sir, next. This is my methodology work. The isocracy species, the different condition and nitrogen and phosphorus induction. These are the different conditions from the nitrogen deprivation and the repletion condition. The biomass, the biochemical estimations, and the gene expression studies, the lipid production by the FAME and GCMS analysis. The gene expression studies was the RNA extraction and cDNA construction and semi-quantity PCR analysis. Next, coming to the cell growth and biomass. The cell growth was the production from the nitrogen repletion and depletion condition from the day 7. The biomass and the nitrogen repletion and depletion from the 7th day. The chlorophyll and the protein content was the highest production from the nitrogen repletion and depletion condition on the day 7. The protein and the nitrogen, the repletion and depletion condition from the day 7. This is the carotenoid and carbohydrate and the lipid production. The total carotenoid productions was the marine microalgae isocracy species was the nitrogen depletion conditions was the highest production from the day 7. The carbohydrate, the highest production from the day 7 for 7. The total lipid and the total lipid content for the nitrogen depletion condition, the day 7 was the highest production in the marine microalgae isocracy species. Next. The saturated fatty acids and the monounsaturated fatty acids from the marine microalgae isocracy species, the highest found in the nitrogen depletion and repletion condition from the day 7, and the stearic acid, the production of the nitrogen depletion condition only. The oleic acids, they move up from the isocracy species, the highest production from the nitrogen depletion condition. Next. The linoleic acid and the compoundic acids from the isocracy species, the nitrogen depletion conditions, the highest production from the linoleic acid, and the compoundic acid, the nitrogen depletion condition from day 7. Next. The alpha-linoleic acid, the nitrogen depletion condition, the highest found in from alpha-linoleic acid, the omega-3 fatty acids from EPA, the nitrogen depletion condition, the third day. The THA is the nitrogen depletion condition from the highest production for the omega-3 fatty acids. Next.
Excuse me, sir. Your time is up, sir. Kindly conclude your presentation, sir.
Next. Wait. These are the semi-quantity analysis PCR, the expression from the desaccharide genes. This is the molecular, the another part of work. This is a molecular work from the band intensity from the gel electrophoresis. The nitrogen depletion condition for highest production from the delta 6 genes. Next slide. The expression the delta 5. This is the nitrogen depletion condition from the EPA condition production to do the marine microalgae isochrosis species. Next. This is a delta 4 genes, the expression from the nitrogen depletion condition, the day 7, the production of the highly found in the THC production. This is a correlation study from the GC analysis from the molecular level for the expression of marine microalgae species. This is summary. In both nitrogen and phosphorus depletion condition, the highest cell density and biomass were observed. The results showed that the nitrogen depletion increased the lipid content than they control. The nitrogen depletion conditions specifically the upregulated from delta 6 TCG genes, the expression and thereby the increased GLA production. The nitrogen depletion increased the delta 5 genes expression and thereby increased by the EPA production. The nitrogen depletion and also effectively the upregulated from the expression of delta 6 TCG and increased the production of the THC when compared with the control cells. Next. Thank you for this. That people are my collaborators. I am under working from Dr. Professor Pradeep, the IIT Madras. He are awarded from the Sri Vigyan Award, the 2025. Another person is Dr. Ben Murugan. He is my senior. So he is working in the KMC's Medical College and Hospital. This is Professor Dr. Gadiresan. He is working from the CUTN, the Professor of Heat and Biotechnology Department. Thank you to all.
Thank you, Dr. Parthasarathi. So you are increasing the production of fatty acid in microalgae.
Yes.
Okay. So how we are introducing gene or what?
Only for the induction from the nitrogen starvation condition. So this is my part of work. Okay. The plant growth regulator and the inhibitors are the workings from here.
It's in which condition? In vitro or it is in that on-field condition or in vitro condition?
Only for lab conditions.
Lab conditions.
Lab conditions.
Okay. Thank you.
Thank you.
Judges, kindly note that we are going to switch over to the online presentations. And first on the presentation mode is lot number 11. Lot number 11.
Ashita, you can present your presentation, ma'am.
Am I audible, ma'am?
Yes, you're audible. You can proceed.
Ma'am, am I audible? I can't hear.
Isma, you're audible. You can proceed.
Hello.
Ashita, you can
Yes,
share
ma'am.
your
Am I
screen.
audible now?
You can proceed
Okay, okay, thank
with your presentation.
you. Okay, thank you. Is my screen visible? Hello.
Your
Is my
screen is visible. You can present.
Okay. Good afternoon, everyone. Myself, Ashita Lanson, Research Scholar, Department of Botany, University College, Trivandrum. Today, I'm going to present my research findings on the title Phytochemical Profiling of Pyrosia heterophylla and Pyrosia pilocellaris from Kerala, a comparative study using HPTLC and HRLCMS QTOF analysis. Genus Pyrosia is an epiphytic fern genus in the family Polypodiaceae. They are found on the tropical and temperate regions. They are commonly known as fell ferns with the presence of stellate hairs and wool-like hairs covering the lamina. Simple leaves with stellate hairs are the distinguishing trait for the genus Pyrosia. 65 species were reported worldwide and 23 species were reported from India. Among the 23 species, 4 species, that is Pyrosia heterophylla, Pyrosia lanceolata, Pyrosia porosa, and Pyrosia ceylanica, were reported from South India. Among them, Pyrosia pilocellaris is rediscovered from Kerala and confirms its existence in South India during our study. Pyrosia heterophylla and Pyrosia pilocellaris exhibit striking morphological similarities, often leading to challenges in taxonomic discrimination. Objectives of the study: to establish a comprehensive chemical fingerprint for both species, to identify and compare secondary metabolites through HRLCMS QTOF analysis, to distinguish morpho-anatomical similar species through metabolic profiling for quality control in pharmacological applications. Materials and methods in HPTLC analysis were carried out through sample preparation and application, documentation, and post-chromatographic derivatization. The methanolic extracts were applied on different tracks on silica gel pre-coated aluminum sheet, and the mobile phase used were toluene ethyl acetate in the ratio 5:1. The developed chromatogram was air dried and visualized, and the images were captured under UV light at 254 nanometer and 366 nanometer, and the fingerprint profiles were documented. Then the plate was derivatized using balanced sulfuric acid reagent and scanned at 575 nanometer, and the RF values and the fingerprint data were documented. The HRLCMS QTOF analysis was conducted on Agilent 1200 series HPLC system equipped with a Thermos Betasil C18 analytical column coupled to an Agilent 6520 QTOF HRMS fitted with an ESI interface. Results of analysis. HPTLC analysis performed on the methanolic extract of Pyrosia heterophylla and Pyrosia pilocellaris showed various chromatogram peaks. The number of peaks indicate the presence of different phytoconstituents present in the sample. The RF values calculated for the phytoconstituents present in the sample would also be helpful in the identification of unknown compounds. Chemotaxonomic differentiation between species is also possible. The results of HPTLC analysis of Pyrosia heterophylla showed major peaks at 254 nanometer, in which peak 7 had highest area percentage of 21.42. 11 major peaks at 366 nanometer, and which peak 11 had highest area percentage of 50.32. 10 major peaks at 545 nanometer, in which peak 5 had highest area percentage of 30.32. Coming to the pilocellaris, 6 major peaks at 254 nanometer, in which peak 5 had highest area percentage of 25.58. 11 major peaks at 366 nanometer, and 5 highest area percentage of 74.64, and 8 major peaks at 545 nanometer, in which peak 10 had highest area percentage of 36.02. The table shows the results of HRLCMS QTOF analysis. The first table is the chemical profile of Pyrosia heterophylla. The major compounds identified were 6-acetyl 7 rhamnoside, dodecanoic acid, bornyl butyrate, 3-hydroxyl coumarin, 16-hydroxyl 10-oxodecanoic acid, quincasiol, 3,3,5-trihydroxyl 4,7-dimethoxyflavonol, catechin, and pedalin. And the next table shows the chemical profile of Pyrosia pilocelluloides, and the common compounds identified were dihydrocaffeic acid 3-ogluconide, butadienol glycoside, myrcetin 7 rhamnoside, bruceatin, 7,7-dimethyl 2-methylene bicycloheptane, octadecyl fumarate, caffeic acid, malic acid, lancerin, catalopside, 102-hydroxyl 4-methoxycinnamyl BD-glucose, and anoglabacin A. And this table shows the common compounds in both the species, which includes quercetin, artemethin, palmitic acid, 7,14-bisbolene, 2-10 tetrol, euphorine, methyl 2-furoate, resorcerols, caparin 2-glucoside, cyanarosate, calisanthe. The HRLC Institute of Analysis detected the presence of flavonoid, fatty acid, terpenoid, phenolics, alkaloid, carboxylic ester, and flavonoid or glycoside compounds in both the Pyrosia species, and in Pyrosia heterophylla showed the presence of flavonoid, fatty acid, terpenoid, and glycosidic compounds. The Pyrosia pilocelluloides reveals the presence of phenolic compounds, terpenoids, glycosides, flavonoids, and flavonoid or glycosidic compounds. Summary and conclusion. In the present study, the comparative metabolic profiling of Pyrosia heterophylla and Pyrosia pilocelluloides were carried out using HPTL, HRLCMS, QTOF analysis. Several primary and secondary metabolites were identified, and significant qualitative differences noticed between the species. The comprehensive chemical profiling using these techniques make it possible to distinguish these species based on the metabolic profile along with the morphoanatomical and also the applications. These are the references used in the study, and thank you. Ma'am? Hello, ma'am?
Ashita?
Yes, ma'am.
Thank you for your excellent presentation, ma. Now, for the time being, you can leave the meeting or you can stay back. We will move on to the next lot, number 12. Ms. Kavya Anjali, are you ready with your presentation?
Yes, ma'am. Good morning, ma'am.
Yeah, you can share your screen and present, ma.
Yes, ma'am. I'm start sharing, ma'am. 1 minute, ma'am.
Ms. Kavya Anjali, please share your screen and present, ma.
Yes, ma'am. 1 minute, ma'am.
If you have any difficulty, we'll move on to the next lot. You can present after the next lot. Is it okay for you? Ms. D.S. Abhinaya, are you ready with your presentation? Ms. D.S. Abhinaya, kindly unmute and answer me. Okay, I'll move on to the next lot. Dr. Reshpi Gopalakrishnan.
Hello, ma'am. Sorry, ma'am. Sorry for the... Hello, ma'am.
Yeah, who is speaking?
Kavya, ma'am.
Okay,
Sorry,
then
ma'am.
now you can proceed, ma'am. We can see your screen. You can proceed.
Yes, ma'am.
Okay, ma'am.
Good morning, ma'am. I'm Kavya Anjali from Justice Bashir Ahmed Sai College for Women. I'm presenting on the topic of Bengal gram as a cost-effective nitrogen substitute for the optimized growth of probiotic lactobacillus species. The aim is to, in microbiology, for culturing, the media is very important. In probiotic industry, lactobacillus is widely used probiotic genus. So I'm trying to make the media in a cost-effective manner, ma'am. So to develop and evaluate cost-effective plant-based fermentation medium, expensive commercial peptone and tryptone is replaced with Bengal gram as a nitrogen source for the optimal growth of probiotic lactobacillus species. The objective of this study is to formulate the cost-effective media and to quantify the soluble protein present in the Bengal gram powder and evaluate the growth performance of lactobacillus species in the modified medium. For that, control used as LB medium and nutrient medium. The LB medium is nothing but lysogeny broth. To assess the efficacy of Bengal gram, the nutritional requirements of fastidious growth. After sticking, the 24 and 48 hours has been growth performance has been checked, ma'am. And the introduction is the lactobacillus is a species or industrially significant probiotic. It widely used in food and pharmaceutical sectors, and these are ecologically and metabolically fastidious bacteria. And it necessity of the growth of these species is nitrogen source. For that, I used Bengal gram as a nitrogen source. That is a traditional microbiological medium. Traditionally, the media expensively contain animal-derived peptones and tryptones. So I am used plant-based peptones and plant-derived nitrogen source. Next, the Bengal gram presents a viable nutrient-rich alternative, and this research evaluate the efficiency of Bengal gram as a lowest cost medium. And the materials are the lysogeny broth and nutrient agar medium is used as control. Bengal gram is a sample. It has a nitrogen source. The all medium is a common components are beef extract, agar, and sodium chloride. For protein estimation in Bengal gram, how much of protein content is to estimate that protein in Bengal gram, Lowry's method has been followed. The other solvent medium microbial culture is used as lactobacillus culture. The methodology has been followed as the Bengal gram seed. In Tamil, it has been pronounced as kadala paruppu. It has been taken, it is grind into powder. From that, 1 gram was taken and estimated for protein. After that, media preparation has been done. The lysogeny medium and nutrient medium is commonly available. From that, sample preparation Bengal gram has been followed that same procedure. Peptone has been replaced with Bengal gram powder. The media is prepared. After that, the sterilization and plate pouring was done. And in that medium, lactobacillus was treated. After that, incubation and growth performance has been done, ma'am. 1 minute, ma'am. Sorry. Sorry, ma'am. And results, the protein estimation, the Lowry's method tabular column this is standard curve of protein, Bengal gram protein estimation. This represents the concentration of the protein present in 1 gram of Bengal gram powder, 0.25 mg. After that, this is the comparison. The basal medium, the nitrogen source in LB media is tryptone and nutrient agar. Replacing this nitrogen source, I used Bengal gram powder. These both are used as LB medium and nutrient medium is used as a control, and this used as a sample. Before 24 hours, there is a moderate growth, and after 48 hours, there is a heavy growth. of Lactobacillus species. This is a culture medium, and this is 24 hours growth picture, and this is 48 hours growth picture, ma'am. After that, the discussion, the protein content of Bengal gram estimated by Lowry's method, 0.25 per gram, confirming its high protein density, and lower Lowry's assay well suited for plants due to its sensitivity, peptide bond, and aromatic acid. This value agrees with recent studies present in chickpea protein content, range 20 to 30%. So this quantified level of protein support the use of Bengal gram as an effective alternative nitrogen source for microbial cultivation. For the growth of bacillus species observed at 24 and 48 hours in the Bengal gram based medium, comparable with the nutrient agar medium and lysogeny broth medium. It effectively utilized as a plant derived nitrogen source. So this consists biomass observed over both in period. Bengal gram can successfully replace peptone and tryptone without compromising microbial growth efficiency. And the conclusion is it contains high level protein. It successfully replaced peptone and tryptone, and lactobacillus growth of 24 and 48 hours as compared to the standard medias. Microbial growth was not optimized. It's cost effective and sustainable alternative for probiotic media and fermentation industry for cost effective media production. The future prospect is optimized for large scale production. In the future, the microbial characterization and biochemical characterization has to be done. For the shelf life and antimicrobial activity has to be done. So final is the approach supports sustainable and low cost media development for the fermentation industry. References, and thank you, ma'am. Thank you for your patience. Ma'am?
Thank you for your wonderful presentation, Ma. You just stay back. You mute yourself and stay back. We'll move to the next presentation. Sri Rajalakshmi, are you ready with your presentation? Sri Rajalakshmi. Are you ready with your presentation? Can you hear me, Ma? Rajalakshmi. If you're not responding, we shall close the meet. Thank you all for joining online. With this, we conclude this Google Meet. Thank you so much.
Hello.
Thank you.
Good afternoon, ma'am.
Hello. This is
Good afternoon, ma'am. This is Sri Rajalakshmi. Yes, ma'am.
Okay, ma'am. You can share your PPT and
Yes,
you can
ma'am.
proceed your presentation.
Yes, ma'am. Yes.
Okay, ma'am.
Ma'am, shall I start?
Yes, you can proceed, ma.
Good afternoon, one and all. This is Sri Rajalakshmi. I am a part-time research scholar, MR Government Arts and Science College, Mannargudi, and I am doing my research under the guidance of Dr. K. Pannirselvam sir, Research Supervisor,
PG and Research Department of Microbiology, MR Government Arts College, Manargudi. And my topic today is regional distribution and prevalence of virulent and hypervirulent Klebsiella pneumoniae post-COVID era, a cross-sectional study in and around Madurai. In recent years, there has been a noticeable increase in infection caused by Klebsiella pneumoniae, particularly due to virulent and hypervirulent strains. Understanding their regional distribution and prevalence is essential for early diagnosis, infection control, and appropriate treatment strategies. With this background, I conducted a cross-sectional study in and around Madurai. Now I move on to my synopsis. Let me briefly present the synopsis of my work. This is my significance of the present study. Klebsiella pneumonia reported among neonates, children, adults, as well as among elderly, as it causes wide range of nosocomial as well as community-associated infections such as UTI, pneumonia, and bacteremia. Injudicious use of antibiotics among patients has led to the emergence of multi-drug-resistant Klebsiella pneumoniae, causing serious opportunistic infection. Pathogenic Klebsiella pneumoniae harbor multiple types of virulent gene, causing outbreaks in hospitals and pose a challenge to treatment due to acquired multi-drug resistant and virulence factors. The increased incidence of hypermucoviscous strains further complicates the microbiological cure of Klebsiella pneumoniae infections. ESBL Klebsiella pneumoniae is commonly reported from Indian hospitals, as it is important to prevent resistant bacteria by correctly identifying ESBL producing Klebsiella pneumoniae and treating infected patients with appropriate antibiotics, and hence hospital infection controls plays a critical role to curb outbreaks. And here are all my objectives: to determine the prevalence of Klebsiella pneumoniae from the clinical samples collected from 3 districts such as Madurai, Sivagangai, and Dindikal, to identify regional distribution patterns and compare the occurrence of virulent and hypervirulent strains in the study area, to differentiate, and my third objective, to differentiate virulent and hypervirulent strains, and to identify the phenotypic screening of virulence and hypervirulence factors among the selected isolates of the test Klebsiella pneumoniae. And my study was carried out after getting the ethical clearance at Institutional Ethics Committee at Arthur Ashirvadam Hospitals, Madurai. These are all my research gap. I have reviewed many papers. Due to insufficient time, I can display some papers here. According to Thomas et al., in their study entitled Hypervirulent Klebsiella pneumoniae, published in eBiomedicine, and their authors' observations are the evidence that hypomucoidity and siderophore production plays an important role in the hypervirulent phenotype, but experimental studies have typically focused only on a single strain, resulting in variable outcomes and a lack of clarity. Hence, need the gold standard experimental approach for distinguishing hypervirulent from classic strains. And my second author, I have reviewed Thomas A. Russo, entitled the paper Klebsiella pneumoniae hypervirulent, the next superbug, published in Future Microbiology, and the authors observed, or the virulence of the isolate was compared with 4 classical Klebsiella pneumoniae blood isolates, and hypervirulent Klebsiella pneumoniae was more resistant to complement and neutrophil mediated suicidal activity. Finally, undoubtedly, many cases of hypervirulent Klebsiella pneumoniae infection are presently unrecognized. And the third paper, I have reviewed clinical and laboratory insights into the threat of hypervirulent Klebsiella pneumoniae, and the author is Zheng Hong et al., published in September 2024, and the author observations are differential diagnosis and early identification of hypervirulent Klebsiella pneumoniae strains are crucial for effective treatment patient care, and the implementation of appropriate infection control measures combining genetic markers and virulence phenotype assays is essential for accurate hypervirulent Klebsiella pneumoniae assessment. Exploring alternative treatment strategies such as therapy is necessary to address highlighting the hypervirulent Klebsiella pneumoniae antibiotic resistance. And my next author is Thomas A. Russo et al., entitled Hypervirulent Klebsiella pneumoniae. And the observations are hypervirulent Klebsiella pneumoniae is more virulent than classical Klebsiella pneumoniae, and a definitive diagnosis requires identification of specific biomarkers, but assays for these markers are not presently FDA approved or routinely performed by clinical microbiology laboratories. Implementing infection control measures when a hospitalized patient is infected with an hypervirulent Klebsiella pneumoniae. Now move on to virulent and hypervirulent Klebsiella pneumoniae. Hypervirulent Klebsiella pneumoniae are more virulent than classical Klebsiella pneumoniae. Humans are the primary carriers of Klebsiella pneumoniae in the environment. People with weakened immune system due to medications or medical condition have a higher risk. Recovery depends on several factors including your age, health status, type of infection, severity of infection, etc. Increased capsule protection that protects the bacteria from phagocytosis, siderophores, or another virulence factor that is needed by the organism to cause infection in hosts. These are the difference between the classical and hypervirulent Klebsiella pneumoniae. Classical Klebsiella pneumoniae choose less capsule protection. In case of hypervirulent Klebsiella pneumoniae, having more capsule protection. And the string test, string is less than 5 mm, and in case of hypervirulent is more than 5 mm. And classical Klebsiella pneumoniae causes infection to immunocompromised individuals and elderly patients. In case of hypervirulent Klebsiella pneumoniae, it causes infection to healthy individuals also. And classical Klebsiella pneumoniae, the virulence factors are siderophore protection, and hypervirulent Klebsiella pneumoniae, siderophore protection, and also the string test. These are all my methodologies, and my exclusive criteria are post-COVID patients except febrile fits, heart disease, rheumatoid arthritis, diabetes, blood pressure, hyperlipidemia, and my inclusive criteria of my study is post-COVID patients with present illness of fever, UTI, bronchitis, abdominal discomfort, throat sore, bacteremia, and sepsis. Collection and analysis of data. These specimens are collected from 3 districts.
Sorry to interrupt, Rajalakshmi. Yeah, you're exceeding the time limit. Can you please conclude this with a summary?
Yeah, yes, ma'am.
Please, thank you.
The test for hypervirulent Klebsiella pneumoniae is the string test. String test is nothing but hypermucoviscosity was defined by the formation of viscous strings 5 mm greater than 5 mm in length when a loop was used to stretch the colony on agar plate is known as positive string test. And my results, I have recruited totally 254 Klebsiella pneumoniae isolates during the period from July 2022 to June 2026 for 2 years. Total virulent Klebsiella pneumoniae I have isolated is 204, and total hypervirulent Klebsiella pneumoniae is 50 cases. wise distribution, 37.4% of 26 to 45 age group was infected more. And based on my sex-wise distribution, females are highly infected with 63.2%. Based on district-wise distribution, Madurai tops the first with 58.7%. And based on wise distribution, I have isolated more number of Klebsiella pneumoniae in urine specimen. And based on vaccination, 90.6% were vaccinated for COVID and 9.4% were non-vaccinated. And diabetic history, 68.9% were non-diabetic and 31.1% were diabetic. Based on blood pressure history, 47.6 had hypertension and 52.4 had not BP. And my overall findings, the total cases recorded from 1st July 2022 to 30 June 2025 is 254, and the higher infections were found in the age group of 26 to 45 with 37.4%, and females were highly infected with 63.2%. Madurai district recorded higher cases when other two districts of Sivagangai and Dindikal with 58.7%. Based on specimen-wise distribution, 59% of samples were collected and 24.8% pus samples were collected. Based on vaccination history, 90.6% were vaccinated for COVID and 9.4% were non-vaccinated. The percentage of isolation of Klebsiella pneumoniae from the post-COVID cases is high, even though vaccination. Hence proved that vaccination is not an effective for secondary bacterial infection. The major reason for bacterial infection in younger adults is those infected with comorbid conditions. In addition, old age adults and children with immunocompromise is also a major reason for more number of Klebsiella
Sri
pneumoniae.
Rajalakshmi, we have already informed that you are exceeding the time limit. Still, you are moving on with your
Yeah, it's
presentation.
over, ma'am.
Okay.
Final slide, ma'am. It's now current antibiotic regimen, novel antibiotics. When ESBL is diagnosed in Klebsiella pneumoniae, carbapenem therapy should be initiated due to its rate of sensitivity across the globe. ESBL producing strains of Klebsiella pneumoniae were detected by ceftazidime and clavulanic acid, and increased diameter of more than 5 mm with ceftazidime plus clavulanic acid as compared to normal ceftazidime disc was considered as positive for ESBL detection. Some superbug strains of Klebsiella pneumoniae are resistant to most antibiotics, including carbapenem, which are considered as a last resort of drugs. Hence, currently using combination of the antibiotics colistin, meropenem, and tetracycline. These are all my references, and thank you so much. Thank you, ma'am.
Now it's time to show our gratitude to judge this section on oral presentation. Dr. L. Devasri ma'am, Head of the Department, Zoology, the Madura College. Dr. S. Karpasamy sir, Deputy Controller of Examination, College. May I kindly request Dr. N. Paneerselvam sir to present a memento of gratitude to our honorable judge. Dr. Thank you, sir. Thank you, ma'am. now it's time for lunch. Lunch will be served in front of our department. After the lunch break, you will assemble around 2:45 in this hall. Kindly be here by 5. Thank you, students. participants, those who are standing outside the hall, kindly come inside and take your seats. Please occupy your seats, please. Priyadarshini, Vindhya. Mudila Chami. Volunteers. போய் எல்லாரும் கூட்டிட்டு வாமா போமா நீங்க செல்போன்லயே இருக்காதீங்க. Dear participants, those who are sitting outside the hall, occupy your seat. Second year sir, எல்லா கூப்பிட்டு உள்ள வர சொல்லுப்பா. அவர் சிசி 3 வரேன்றார் மேடம். ஓகே சார். குமார் அண்ணா இது வேணாம், இது இங்க தான் கேட்கும், அங்க கேட்காது. வெளியில kind request participants, those who are sitting out, enter the conference hall so that we can start immediately and finish it as soon as possible. I request all the students to come inside. Kindly cooperate, students. Please come and sit inside the conference hall. சார்ட்ட சொல்லிட்டு போயிடுங்க சார். இதை பார்க்க சரி நீ மட்டும் உள்ள போங்க போங்க உள்ள. I kindly request the students and the participants of microbiology department to assemble immediately. சார் அங்கே யார் சார் இருக்காங்க? Dear participants, please assemble immediately. Third year students, please come and assemble immediately. Hurry. Okay? இதுக்கு மேல என்னால பண்ண போதும் மேடம், இவ்வளவு கூட்டம் போதும். Jayashree of MSc, Jayashree of second MSc, kindly. Jayashree, where is Jayashree? Please, come here. Come here. Let's start the session. Start the session. In the Ridane. I kindly request the students, those who are standing outside, please assemble. We are going to start the session.
Respected chief guests, distinguished speakers, dear participants, very good afternoon. As every meaningful academic journey reaches a moment of reflection and gratitude, we now proceed to the valedictory session of the conference. This session marks not only the conclusion of the event, but shared collaboration and intellectual enrichment achieved over the course of the conference. Introducing the speaker, formally commence the valedictory session, it is my privilege to invite Dr. P. Rajan sir to introduce the valedictory.
Good afternoon, everyone. It's my honor to introduce our distinguished speaker, Dr. M.R. Vasan, Madura Medical College. Completed MBS in Government Mohan Kumaramangalam Medical College, Salem, and MD post-graduation at Government Kilpak Medical College. Sir received and best outgoing student in MD Microbiology and post-graduation. Currently, Sir is working as associate at the Institute of Microbiology, Madurai Medical College. Sir has 14 years of teaching experience for undergraduate and postgraduate and paramedical courses. And Sir has published an enormous number of national and international journals. Under Sir's leadership, TB Diagnostic Laboratory accredited for first line and second line TB drugs by line probe assay testing. And also Sir underwent the training in ISO 1592022 for HIV viral load laboratories. Sir also serve as a product for dental and pharmacy. He has written for MD post also. During COVID pandemic, Sir served as co-principal investigator in VRDL laboratory. Team processed more than 28 lakh sample, that was the highest in Tamil Nadu. Sir received best doctor from district collector also. So we welcome Dr. Vasant sir to share his insights on diagnostic transition in infection.
Felicitating the speaker by Dr. Selvam sir.
SDA.
Felicitating the speaker by Dr. Selvam sir. can start.
அப்படின்னா இது கனெக்ட் ஆகலையா? ஒரு ஃபங்க்ஷன் நடக்குது நமக்கு. சவுண்ட்
Is it possible to detect the liquid only or tissue?
Anything. Any except blood, we can do sample for TB.
Oh, it's possible tissue also?
Tissue also.
Oh.
எல்லாத்துக்கும் தெரியும் பிளட்ல வந்து RBC இருக்கு. RBC PCR for PCR. அதனால நம்ம பிளட்டை தவிர எந்த சாம்பிளும் நீங்க சிபி நேட்ல ரன் பண்ணலாம். ஸோ டிஷ்யூ ரன் பண்ணலாம், போன் ரன் பண்ணலாம், யூரின் ரன் பண்ணலாம், CSF, ஸ்பியூட்டம், எனி டிஷ்யூ எது வேணாலும் நீங்கள் ரன் பண்ணலாம். ஈவன் we can run CB net.
Sir, last question. We have the lots of in India and also government of Tamil Nadu. We have the institute. So we are facing tuberculosis. Why? Because government of India, government of Tamil Nadu deliberate the awareness. Still we are facing.
It's two reasons, sir.
Experts, so
yes, so two reasons for the TB is still existing. Number one, the treatment is not completed by the patients. Number one.
Okay.
Number two, the document or notification. If the patient TB, he and get the medicine and he is not intimating to the government, then until they are having the money, the government is giving the free of cost. Along with the government is giving every month 1,000 rupees for their nutritional support, but they are not notifying to the government. So notification is lagging. Number two, they are taking the medicine for not completing course. So ஏன்னா TB drug எடுத்தீங்கன்னா டெஃபினட்டாக சைடு எஃபெக்ட் இருக்கும். ஸோ அந்த சைடு எஃபெக்ட்னால என்ன பண்ணுறாங்கன்னா ஒரு 2 months எடுப்பாங்க. ஸோ தே வில் ட்ரீட்மெண்ட். பட் கம்ப்ளீட்டாக 6 months எடுத்தால் தான் ஆர்கானிசம் வில் கம்ப்ளீட்லி கிளியர். அதர்வைஸ் ஆர்கானிசம் இஸ் கெட்டிங் ரெசிஸ்டன்ஸ். அந்த ரெசிஸ்டன்ஸுக்கு நம்ம கிட்ட ட்ரீட்மெண்ட் நாட் அவைலபிள்.
ஓகே,
தட் இஸ் த மெயின் ரீசன்.
தேங்க்யூ ஸோ மச் சார்.
தேங்க்யூ சார்.
Any other clarification needed? If there is no, we'll proceed.
Okay. So thank you for patient listening.
Thank you, sir, for your enlightening and thought-provoking lecture. Vote of thanks by Mr. J. Amala Jaykumar, sir.
Hearty welcome. It is a great honor to propose a vote of thanks to everyone who helped to us make the conference such a great success. I thank our outstanding speaker, Dr. Professor, Madurai Medical College, Madurai, and for making the session exciting and meaningful. I desire to articulate our sincere gratitude to management, principal, chief coordinator, and Department of Biotechnology and Department of Microbiology and non-teaching staff members and for their moral support and the guidance, as no program can become successful with a single person. I sincere thank to all our college students. My sincere thanks to the program convener and the one thank to everyone, and thank you all for making the sessions successful for events. Thank you.
Thank you, sir. We shall now have the conference report presentation summarizing the academic highlights of the conference. I invite Dr. N. Rajesh Kannan sir to kindly present the report.
The International Conference on Molecular Microbiology, Challenges and Opportunities in Era, MMCOPE 2026 was successfully organized with a strong academic. The International Conference on Molecular Microbiology, Challenges and Opportunities in the Post-Pandemic Era, MMCOPE, was successfully organized with a strong academic focus and wide scholarly participation. The conference received a total 111 research abstracts comprising 69 oral presentations and 42 poster presentations, reflecting the relevance and contemporary importance of the conference theme. Abstract submissions were received from reputed universities, medical colleges, and research institutions, highlighting current research trends and advancement in molecular microbiology and related discipline. The scientific sessions promoted meaningful knowledge and interaction among senior scientists, faculty members, and researchers and young scholars. The conference proceedings were published with the ISBN number, ensuring academic authenticity, permanent archival value, and institutional recognition. The ISBN indexed abstract volume support academic referencing and research documentation. The conference was conducted in hybrid with the live streaming through the YouTube, which expanded academic outreach, participation from a broader national and international audience. This digital approach enhanced accessibility and dissemination of scientific knowledge. Overall, MMCOPE 2026 provided a valuable academic platform for sharing research outcomes, strengthening scientific collaboration, and advancing the molecular microbiology in addressing post-pandemic global health challenges. Thank you.
Thank you, sir, for the comprehensive conference report.